and welcome to the podcast of Loughborough University's Institute of Advanced Studies, a podcast where our international fellows discuss the most pressing issues of today that we're all facing and share their exciting research ideas. We hope you enjoy today's talk. Thank you for tuning in.
Many thanks for come and join these seminars. And I'm really feel privileged to invite Vickie, Shim from the Auckland Bioengineering Institute, talking about her work related to the sports brain injury, specifically about rugby player. And I think she has been working with a big team, working in this area for quite a long time. And what we see today is not something, like 5 years work. It's more than, I think, 10 years work, continuous work. I will leave the platform to Vickie. I'm sure at the end there will be loads of questions, but please hold your question till the end. Yeah, and also from the online who joined this seminar, please hold your question to the end. And I'm sure we will have a lot of interactions and discussions. Thank you. Your platform.
Thank you. So thank you, Yang, for this wonderful opportunity to be here at Loughborough as a visiting fellow of IAS. I got here last Wednesday. I enjoyed my week here very thoroughly, and I really envy that how wonderful your campus is. So you guys in a very good place. I can assure you that, because I can show you how Auckland campus is looking like. Oh, that's the point. Yes. Okay. So Auckland University is in Auckland. Don't have a campus. It's just the buildings scattered around the city. So this is our new engineering building, and behind it is our Auckland Bioengineering Institute here. As you can see from, and this is our building. So it was established by Sir Peter Hunter in 2000. Our university has a thing called the Large Scale Research Institutes that mainly does research only. So we are one of the two. And currently there are over 300 staffs and over 100 PhD students. So our major research theme is in computational and experimental biophysiology, studying from heart, lung, GI system, and musculoskeletal system. But today I'll be talking about the work that my group is doing, which is brain biomechanics group. So what do I mean by brain biomechanics? So we are all talking about the sports-related concussion, where players are tackled by opponents or some other means, and they have a head impact either directly or indirectly. So we call it head acceleration events because it does not necessarily involve direct hit to the head. So what is TBI, traumatic brain injury? Like many other neurodegenerative diseases, it is a spectrum. So you can't define like you are this, you are that. You are part of the spectrum here. And as you can see from here, this mTBI is very broad in terms of this TBI spectrum. And underneath mTBI, there are two categories. One is concussion, which also has a bit of acute symptoms. But what is more common is this non-symptomatic subconcussive impact. So we are also looking at this concussion and subconcussive impact. But this term mild might be a little bit misleading, because is it really mild? As you all know, there are this series of media reports in the past that highlighted the dangers of even single concussion that could lead to a heightened risk of Parkinson's disease. Or especially the concern in like Western countries is that the children developing brain, what is the effect of having this concussion or subconcussive impact at early age? So Canada has some movement to restrict the football practices, and the soccer as well, the header impact. And in New Zealand, there was a call from a national newspaper that the children under 12 should not play contact sports at all. So this all because of the fear of unknown. We don't know exactly what kind of effect this concussion and repetitive head impact, subconcussive head impact have on our brain, especially developing brain. So unfortunately, there is no solution, no treatment for concussion or mTBI. More than 30 clinical trials failed completely. And then it is notoriously difficult to diagnose objectively. The current solution is that follow my finger. So they try to see if you follow your finger, if a player or someone with a concussion can follow the finger, then that's it. You can go home. So, but why some go on to experience persistent symptoms while others recover naturally? How do we differentiate these two different cohorts? So that's what we want to do in our study. So we have taken this grassroots approach where we are working very closely with the community to collect real-world concussion data. Some of the questions that we have is how do concussion or repetitive subconcussive impact change your brain? We're using three main modalities. One is advanced MRI, second is instrumented mouthguards, and the third one is subject-specific finite element analysis. So we are collecting rugby data set with Gisborne Boys High School. So just to orient you guys where Gisborne is, this is the map of New Zealand. And Gisborne is located on the east coast of North Island. It's a very scenic, beautiful beachside town where there are only town in the whole world, there are more MRIs than traffic lights because there is only one traffic light in this whole city, but there are two MRI machines. So we've been working with the rugby team there since 2021, followed over 100 players so far. Our study is focused on MRI. So we have taken this advanced MRI images. Usually you probably would have heard of this functional MRI, which shows where in the brain is activated when you are doing a certain task or when you are at rest. This 40 flow shows how the blood flows through your cerebral vasculature. And a structural MRI is, of course, the shape and geometry of the MRI. But here we are focusing on the diffusion MRI. So what is diffusion MRI? So what is diffusion? Diffusion is, in diffusion MRI, we are measuring how the water molecule diffuses within your brain between these white matter fiber structures. So when there is a very jumbled white matter fiber structure, we say the diffusion might look like isotropic diffusion. When there is a clear pathway, it might look anisotropic. So diffusion approximates this movement of this water molecule for every voxel and describes that using this diffusion tensor. And using the diffusion tensor values, we can derive this diffusion matrix, which you can see on your right here. Now, problem with this DTI, diffusion tensor imaging, is that the specificity might suffer a bit. So some parameters like a fractional anisotropy increase or decrease, it may be related to the axonal swelling, demyelination, or other sort of impairment of white matter fiber integrity. However, the problem with the diffusion is that it assumes that the water molecule diffusion in the brain is Gaussian, which is not the case. So there is a new measure called diffusion kurtosis imaging, which assumes that the deviation from Gaussian distribution and give us a better, more dimensional look, high dimensional look of the how motor molecule diffuses within the brain. So in our study, we used both. Now, yeah. Okay. Okay. So how do we analyze this diffusion images? First, we can do a whole brain voxel-wise analysis. This is just called the track-based spatial statistics, where we register all the brains in the common space and derive an average white matter fiber track skeleton, and then do a voxel-wise analysis. That's called TBSS. And another one is tractography, which you might have seen in the pictures, where we can follow the long-range white matter fiber tracks and identify these fibers that connects different parts of the brain. So that's the tractography method that's used in DTI. Another one is identifying the region of interest, such as in this case, the CST track in your brain, corticospinal tract, and then you can identify those regions and analyze the region further. Another one is track profiling, because this region of interest method averages whole region single number. So track profiling methods create a skeleton through the white matter fiber track and then see the variation of these different metrics depending on the location of the white matter fiber track. The last one is, you probably have heard of this, is connectome-based analysis, where it approximates brain as a huge connectome that connects different parts of the brain, and then using the structural connectivity to see how this connection changes due to injury or neurodegenerative diseases. We use all this method in our analysis pipeline. So this is our study design. We recruit the rugby players before the start of the season, and then MRI scan them. And then we scan them again in the mid-season and again in the post-season. And if a player happens to have a concussion, clinically diagnosed concussion, we pull them out and put them through a more rigorous MRI scanning protocol to measure how the acute changes in the brain occurs after the head impact. And then we also scan the control groups at the same time. And our assessment pipeline includes this MRI to start with. And we also have a non-imaging modalities such as neurocognitive testing. We use the SCAT5, and we also measured all the head impacts using this instrumented mouthguard that we deploy to all the players that we measure. So how do we design our study? So we first do the observational study to characterize how microstructural alteration in the brain of contact sports athletes happen. We look at both cross-sectional against the control and longitudinal. And we also correlate these changes in the brain white matter fibers with the non-neuroimaging data. So we use symptom scores, head kinematics, and we also use this head injury criterion or FE analysis as well. And then we are building this machine learning tool that can predict the injury severity and return to play time frame. So that's sort of project overview that we have. So what is the result of this microstructural analysis? As you can see, if you look at the longitudinal changes, we didn't find statistically significant changes. However, when we look at the cross-sectional way, when you compare the changes with the control groups, you can see that there is increase in changes in the white matter fiber tract. Before I go any further, I should orient you this figure. So this green is average white matter fiber tract from everyone. And the red area indicates area that was statistically different, significantly different between these two cohorts. So when you compare to the control, this fractional anisotropy measure, the red area indicates that this area, the control and context-exposed players are different. And then as you can see, as you move from early season to mid-season to post-season, this thing increased. The size of the difference increased. Another thing was this kurtosis measure showed more sensitivity than DTI measures. As you can see, the size of the red area was a lot larger for the kurtosis measures. And these two are the areas that shows significant differences in the white matter fiber tracts. So when we compare with the mTBI group with the control, this is where it was interesting. So significant clusters was formed at 3 time points here for the AK, but it was usual for all 3 measures, the 3 weeks time point was the largest difference. So this might indicate that this is the time point where peak cerebral vulnerability might be occurring. So that's what we have found out. And we also looked at the so those clusters, we further analyzed these red clusters, and then we found out the here you can see that the time point B again was the highest difference for the compared to the control and mTBI group. Interestingly, symptom was highest at time point A, but the imaging shows the highest at time point B. So this might suggest that the symptom resolution may not necessarily mean that brain change has also been completely recovered, because there's difference in the time points for the symptom and the actually white matter fiber tract. And so, and we also correlated, very strong correlation, positive, negative, strong correlation with the various symptom counts and symptom scores. But symptom counts were basically better measure for severity than symptom score. So, in summary, for this part, increasing size of difference between control and cohort suggests that there might be axonal damage or axonal alteration have happened in the brains of those players. And having control was very beneficial for seeing these changes. And then symptom count was a very reliable measure for the correlating this with the changes in the brain. So now we're moving on to this connectivity analysis. So previously it was a TBS whole brain analysis. Now we're moving on to the connectivity analysis. So how do we analyze connection changes in the brain? First, we postulate the brain with the different parts of the brain. There is brain atlases available that you can choose. We chose Desikan-Kiliani atlas. And then we postulate the brain, and then we measure the strength of the connection between regions using this fiber tractography results. And then we make this structural connection between these parts of the brain. That's how the structural connectivity is analyzed in the brain. And we then use our in-house software to derive some graph theory metrics. There are well-known graph theory metrics that measures how efficient the connection between different parts of the brain. And there's a global measure and there's a local measure. And we use the linear mixed model, multiple linear model to see the correlation between these parameters. And we also use what we call NBS connector measure to see how these correlate, how these connections between the cortical regions change due to mTBI or subconcussive head impacts. And we also use the ANOVA to measure those correlation. Okay, so this is the results of structural connectivity analysis. So in terms of global measure, you can see that the global efficiency and clustering coefficients were significantly higher in context-exposed athlete and mTBI at early time points compared to the controls. And in addition, mTBI had a low network density at time point A as well. So for nodal metrics, the had a local efficiency and clustering at this particular region of the brain called left superior frontal cortex, and then for the and the right precentral cortex as well. So this indicates that certain parts of the brain might be affected more than others. The change in global efficiency of these regions. And interestingly, these nodal connection changes were particularly weaker in these regions for the contact sports athletes, which involves hippocampus, and then it involves the what do you call it? Suddenly, it's in the next slide. Frontoparietal network. So there are some changes in the frontoparietal network and the right hippocampus postcentral gyrus here. So our structural connectivity analysis show that the total network connectivity changed. And then in particular, this was particularly interesting, frontoparietal network, because that's the network where executive function is controlled. So for the mTBI, those who had mTBI might have a weaker connection between this frontal parietal network, means their executive function or memory task might be compromised. On the other hand, for context-exposed cohorts, their hippocampus connection was compromised, which means motor sensor connection might be compromised as well. So depending on different type of injury or different type of head impact, different parts of the brain were affected more. So now we're moving on to the looking at the specific track of the brain. So just brief recap. So in the part I just described for DTI method, there are 3 established methods. There is Rowe-based, which is approximating the whole track with a single number. Track profiling is a line, and then TBS as a sheet. But all suffer from loss of information due to averaging. So in the past, we developed a new method called track morphing, where we have this template mesh of the track that you are interested, and then you morph the mesh to match the shape of the subjects. And then you can do subject inter-subject comparison using that method without losing any information. So this is how we generate the geometry shape of the track. So you identify the track that you are interested in. In this particular case, it's a CST track. And then for this one, we have also applied this very complicated track called inferior frontal occipital fasciculus. That's a track that connects front and back of your brain. And because of its long length, it is very well injured from the head impact or movement of your brain. So the way we do it is that we get this shape from the MR tractography, and we create a template 3D mesh, and we use our mesh morphing methods, and then we assign these DTI metrics to all the nodes within the mesh. And then we can do the intra-subject comparison. So we first generate this mesh for all the subjects, and we do a permutation t-test and identify significantly different elements. And we extract those elements and perform principal component analysis to see if they can separate these two groups nicely. So this is the results. As you can see, the number of significant elements increased as the season progressed as well, especially in this region for the body region of the corticospinal tract. You can see the increase in significant elements. And also in this frontal part of the IFOF tract as well. Interestingly, for mTBI, we didn't see the increase, particularly probably because they are taking 2 weeks off. So we don't see any increase in the significantly different elements in these particular cohorts as well. So when we did the PCA on this IFOF track, we were able to differentiate this rugby cohorts against control cohort very nicely. As you can see, there's clear cluster, 2 clusters formed to indicate that these two are quite different, which means that this can be used as a biomarker for detecting brain changes due to contact sports. And also for mTBI cohort, what was interesting was that for different time points, the different color indicates different time points here. So there is not complete separation, but the clusters are formed around different time points, meaning that the brain is at different stage of recovery or getting worse. We are not quite sure which direction. Hopefully it's recovery, but time points, the time-dependent changes in the brain is quite significant in this case. So, in summary of this part, it's a heterogeneous behavior of mTBI group was particularly interesting for us. And then the localization of observed microstructural alteration was also very unique thing people have not observed in the past. So that's the DTI part of the study. So moving on to computational modeling of the brain. So I'll give you a little bit tour of how computational model was used in mTBI. The original model was in the 1970s, where they approximated brain into this very idealized shape. And from 2000 onwards, the models started to have more brain-like shape. And due to advance in the computational technology and computational power, more and more groups started to build more complicated brain models. So for example, I think the KTH model was one of the first that contains all the major components of the brain in the 3D finite element model. And then the Miller in 2016 developed the Atlas-based model where they just turned the voxel into an element. And then this group from Dartmouth was another group that has a very high fidelity model. They can incorporate this white matter fiber track within the model. And then Imperial College model also has a very detailed differentiation between white and gray matter here, as you can see. So so somehow this everybody who developed this kind of previous models got together and wrote a review article. And these are the things that they recommended as a missing. So a lot of models use idealized kinetic inputs. And then what is important in the contact simulation is that how the interface is described. And a lot of models did not use a realistic interface. And the material property is very important, but that was very, some models used the simplified one. And then the validation was quite difficult because it's impossible with the human brain. And lack of efficiency model generation simulation, because usually it takes about 30 hours or few more days to run this one. So as a group, they recommended that the major roadblock for effective real-world user brain model is a lack of sufficient kinematic injury data encompassing sports, sex, and age, and other factors. So we have developed our method where we develop a robust pipeline of generating MRI-based subject-specific fine element model of the brain. And our strength is that we have all the head impact records that player experience throughout the season. So we don't have to have this head impact from the literature. We have the person's own head impact. So we combine it with the player's own impact profile. So we call it a higher brain model. Very, very nice acronym that I'm very proud of that I came up with, which is Head Impact Gisborne Boys High School Elite Rugby Players Brain Model. So this is our model. So how do we generate this subject-specific brain model? We use two different methods. One is the mesh morphing method. Similar to the one that we use for the track morphing, we morph the whole brain using the player's own T1 structural information. And then once the brain is morphed to match the shape of the player, and then we assign for every element in the brain using the DTI FA values. In the past, people have developed for this hyper viscoelastic material property. They found out the correlation between this K parameter in this strain energy density function, and then this is correlated with FA in such a way. So we use this relationship in the literature and assign the different elements to the corresponding different material property, and it resembles very nicely. So top is the element assignment, bottom is the FA map. And we validate our model against the animal study, where we have conducted pre-impact MRI, impact delivery, and then post-impact MRI. And then we simulated that impact delivery situation, and then our strain map was very well matched with the actual observed changes in the brain from MRI. But what was also interesting to us was that the impact level seems to have, the strain level seems to have increased after head impact. Meaning, if the strain is measure of deformation, so strain increase means brain deformed more. That means brain got softer. So some sort of material change is happening in the brain, which we are not quite sure what it is. So we decided to investigate further with the all the head impacts that we measured. This is for 2021 cohorts, where we measured all the head acceleration events for the players during games and practices. And we have all this data curated for the model application. And this is a sort of exemplar subject-specific brain model and strain deformation during a single head impact of 30 milliseconds long. So what we have found is that as I've said, FA change, the DTI metrics change when you compare against the control, right? That's what I said in the previous slides. But when you compare to self, so when you compare to the early season version of your DTI, there is no significant changes in FA or any other diffusion measures. However, when you look at strains, they changed. So this may indicate that strain patterns are more sensitive than MRI alone. So that's what we have found out. And it's not just the magnitude, but also the dynamics. So initially pre-season, your brain oscillated like that. But then mid-season, it oscillated like that. As you can see, not just the magnitude or the frequency changed as well. And the post-season, the dynamics were quite different. So that let us think that obvious that the tissue softening is happening, but can we correlate that, or can we have some sort of measure that can be more correlatable to other imaging biomarkers? So that's why we are trying to bridging brain dynamics and with the emergent tissue properties using a data-driven approach. So as I've shown you, the dynamic strain patterns are very complex. It shows a very rich spatial temporal data, but hard to extract interpretable tissue properties. And there is a data-driven approach called dynamic mode decomposition that identifies modes governing this brain oscillation, dynamic behaviors. Then it can be used to encode physical signatures like frequency and damping. And that's how we get these emergent tissue properties from this strain pattern, such as viscoelastic properties such as shear modulus or damping ratio. So we can, from these results, we can infer effective values we call emergent tissue properties, not because they are not given explicitly in the model. And when we compare the changes in shear modulus and damping ratio, within subject, inter-subject, intra-subject, within season variations were significant. As I said before, DTI was significant when it's cross-sectionally compared with the control, but not when it's compared to its pre-season data set. But shear modulus and damping ratio was quite significantly changed. So that gives us a very strong motivation to apply this to everyone. However, one bottleneck is that to run this, it takes about 30 hours. So it's very numerically intensive methods to run. So the natural progression is to develop brain deformation patterns, predicting brain deformation pattern directly from the sensors by passing this finite element simulation, which is computationally expensive and very time-consuming. So we developed this ML pipeline where we can measure, we can predict brain patterns, strain patterns straight from the sensor data. So how did we do that? So we used a Python library called TS-Fresh, which extracts features from time series data. And once we extract the time series features from the strain and the impact, we can compose a super feature set that mimics, that correlates best from the input to output. And then we use nonlinear regression random forest model to predict the strain values. So our model was I was pretty happy with the performance of this model. So this one, so on your left is the this head acceleration event measured with the wearable sensor, mouthguard sensor. And then this is the resulting strains from the sensor. As you can see, I think this green is not green. Blue is prediction and orange is the target. As you can see that the accuracy is pretty good, and the correlation was pretty good as well, as you can see from this figure. And when we compare to other available machine learning model in the literature, majority of them just predict a single maximum principal strain. But our one predicts the time history of the maximum principal strain, from which we can derive other features like area under curve, which might be a more indicative of brain change than a single number. But when you compare with the other CNN model in the literature, our one outperformed very well. And this is very efficient one too, because this one was generated with about 5,000 impacts. Our one was about 150 impacts. So we are now, so this gives us a very nice way to approximate the brain strain more efficiently, so that we can not only get this strain, but also get the this emergent brain properties, which is more sensitive than the DTI measure only. So, in conclusion, I've shown you that the diffusion MR measures can detect the white matter fiber microstructure changes only after a single season of rugby. And that was with or without concussion. So it was not just the concussion. If you have repetitive subconcussive head impact, chances are your brain might have changed compared to control. And it was very encouraging for us to see that the diffusion metrics, especially the advanced diffusion metrics called the DKI, correlate very highly with the symptom measures through neurocognitive tests, which means that if you have a better designed neurocognitive test, it can be a surrogate for a diagnosis of the concussion or subconcussive head impacts. What was very interesting for us was that the brain strain patterns were more sensitive than either DKI or DTI metrics. Neither of them were able to detect within season intra-subject changes, but strain patterns were different for these within season intra-subject changes. And what was very interesting for us is the emergent material property that we extracted from this 5D strain field were shown statistically significant changes in brain tissue properties within season, which were not possible with any other measures in the MRI models. So we're building this robust and accurate ML pipeline to scale this what we call virtual material property pipeline, and for predicting correlating this with the safe return to time frame, which we believe is one of the best measures that you can correlate to. So I have to thank my students who've done most of the work, basically. So especially Christian and Jacob were, because none of them are here, they did a lot of work. Yeah, let's put it that way. They did lion's share of work, and really all my students worked very hard, and they all did very good work in various different aspects of the project. And all the data was collected at MATI Medical Research Institute in Lisbon, where the team there were instrumental in getting this project going, setting up the relationship with the community groups, which is not a simple task, but they were so good at setting up the relationship and maintaining it. That's why we were able to collect the data for the past 5 years. And we hope we will be able to do that in the future as well, which gives us which is the foundation for all these novel findings that we have in the past. And at the ABI, I work very closely with this our team members at the ABI, and there are some international collaborators, domestic and international collaborators. And funding sources here. Okay, thank you.
Thank you.
Thank you for listening to the podcast of Loughborough University's Institute of Advanced Studies. A quick reminder about how to stay connected. You can find us on Blue Sky, LinkedIn and Instagram, and you can also watch the presentations on YouTube. The podcast is available on all the major platforms. Please download, share, and most importantly, subscribe. See you next time.